exciting world tour that we just had. We're now going to feature some outstanding Canadian talent. And this session is going to be the prelude to a discussion session that's going to happen afterwards in the area where we had the poster session. So please join us, because it'll be a very sort of important way to engage with colleagues here and get a lot of sort of input and maximize benefit of the meeting. So please join us for the discussion afterwards. But to begin, we're going to start with a Canadian who's currently living in America, and we're going to begin with Terry Roemer from Merck. Take it away, Terry.
Is that okay? Yeah. All right. So, okay. Yeah, I too would like to thank Joe and Melissa for the opportunity to come to this summit. It's been a great day. I've really enjoyed myself, and I've met many of you. So. What I was going to do is, I've got this right, is sort of touch on some work that we published really just a couple of weeks ago, and first highlight acknowledge my colleagues. What? This does work. A large number of colleagues. I can't call out all of them, but I will call out a few of them. Who really led some key experiments. And so this work has been published in a brand new journal, ACS Infectious Diseases. I think it's going to be an exciting new journal for all of us. And I think rather than going deep into the technical details of what my title is, what I'd rather do is use this work as sort of an example of the way we think and the strategies that we employ at Merck in antimicrobial lead discovery, and in this case antifungals in particular. So really what I'd like to do is start by just giving you, for what it's worth, a personal perspective on what I believe, and many of us believe at Merck, are some of the guiding principles for hit and lead finding for novel antimicrobials. And I may be provocative here, but I'm going to start out with this, that the way we think is compound-centric, not target-specific, okay? So really what we're trying to do is develop genome-wide whole cell assays that ostensibly screen all targets in the relevant pathogen, again, in a whole cell context, rather than I'm sorry, but the endless debate of what's the best target and why. And with such a strategy, really put the value on the chemical matter, not the target, okay? So start with annotating a library of compounds, identify that small privileged subset that's bioactive and actually has potentially drug-like features like whole cell activity and spectrum, chemically tractable, and link that privileged chemical matter back to target in an agnostic way. And if you think about it, what we're really trying to do then is use chemical matter as probes to empirically identify druggable targets in the genome, right? And then the value there is that having identified a truly druggable target, we actually have chemical matter in hand at time zero. And so the real challenge is, okay, do this iteratively, multiple times. And each time you identify a new compound and a new target, we call those a compound target pair. And if we're to prioritize where we start, we prioritize according to the biology of the target and the chemical tractability of the cognate inhibitor to that target. That's where we begin to prioritize. So I think we all appreciate that natural products, historically have proven to be the place where antimicrobials come from. And synthetic chemistry, there aren't that many examples. And so one of the other sort of principles that we employ is therefore to screen natural products. We've had a gap for 4 or 5 years at Merck. We are getting back into that, and I'm really excited about that. But if you're going to screen natural product crude extracts, you really have to understand the complexity of natural product extracts and their limitations in screening complex mixtures, and sort of mitigate that as best you can up front. You have to have a clever strategy to deal with such issues of rediscovery and dereplication of known antimicrobials in natural product mixtures. Another sort of central principle for us is to have really rapid and robust MOA tools to really unequivocally demonstrate mechanism of action. And an approach that is so simple and so powerful, and I'll touch on, is old-fashioned genetics, but with a twist, where in this case our fungal pathogen is haploid, and we isolate multiple stable, independently derived drug-resistant mutants, and we map those mutations that are causal for the drug resistance by whole genome sequencing. I call that genetic perfection. Where possible, if you think about MOA, normally, we think about this in terms of scoring, terminal phenotypes or chemotypes on a Petri plate. But in an ideal world, wherever possible, if there are added bonuses to that mechanism of action in a whole cell context, in an infectious model setting, where you can actually leverage perhaps what's left of the innate immune response, for example, that's added value, and so we look for that. What is paramount is medicinal chemistry. So I think that, as a biologist, those first 3 bits, I think we got that covered. But that only gets you to hits and leads. And real drug-like opportunities really require, medicinal chemists, pharmacologists. They drive drug development, not us. And, the last point I would make is, all of these points I think are necessary, but they're not sufficient. If you look at the last bullet, right? You have to be incredibly lucky, blessed to actually really score at this. It's that difficult. Okay, so what is our screening paradigm? This is something that we developed many years ago, and it was really modeled based on the pioneering work of the Saccharomyces cerevisiae community, led by Ron Davis, and then really a team effort by some 16 different labs that built the screening resource. So we call it. So we basically copied this approach in Candida albicans. So obviously a medically important fungal pathogen, and it's diploid. And so the first principle that's employed in this screening approach is that of gene dosage. That gene dosage affects chemical sensitivity. And we all know what that really means, right? So you knock down the expression of a particular target. Those cells, that strain, is intrinsically more sensitive to inhibitors of that target simply because there's less of it there in those cells versus other mutants or other strains depleted of other targets or your isogenic parent. So that's the first principle. And so, yeah, so the way we've done this is take advantage of the fact that Candida is diploid and precisely knock out one of the two alleles. So knockdown expression 50%. And we've done this, or we did this some time ago, in a massive scale. So one lab, one group, 12.5 people in Montreal, Quebec, Canada. 5,400 precisely constructed heterozygote deletion mutants. So spanning around 90% genome coverage of the pathogen. The other thing we did is we introduced barcodes before and after the deletion mutation. And so these barcodes are, unique DNA sequences that identify one heterozygote from any of the other heterozygotes, okay? And this is important because it affords us the ability to multiplex, take all of these heterozygote deletion mutants and screen them together in co-culture, right, in a single tube, in a single well. And the idea is to take a compound or a natural product extract which is appealing based on its bioactivity, but you don't know its mechanism of action, okay? And at a sub-MIC, screen that pool and ask if there are any of these heterozygote strains, or as we would call them, a whole cell target-based assay, if any of those strains are uniquely hypersensitive to the compound and would deplete versus a mock treatment over 20 generations or so. And then using the barcodes, whether it's microarrays, whether it's whole genome sequencing, you can de-replicate which of any of these strains have depleted. And the idea, of course, is that any of those strains that have a reduced growth rate, a fitness cost in the presence of the bioactive compound, knowing what those strains are provides very insightful hypotheses as to the mechanism of action. So on the right is just a simple example. I'm not going to go through it. You guys all know about fluconazole. You know the mechanism of action. And you also know mechanisms that are relevant in the clinic. And so this is just one example with fluconazole, and you get this really beautiful, simple profile that captures mechanism of action and mechanism of resistance, right? High resolution. Okay, so let's turn to this paper that I'm supposed to talk about. One of the things we did is we screened roughly 1,000 compounds, 1,000 synthetic small molecules that were bioactive against Candida albicans and can be triaged in other ways to warrant. Take a peek. Can you figure out what the mechanism is? And so this is one example. Compound is called G884. And remember, there are 5,400 heterozygotes that are being assayed in parallel. I'm not showing you 5,400. I'm showing you 7 strains amongst the entire compendium of possible targets that reproducibly demonstrate an altered susceptibility to the small molecule. So you can see here that the most noticeable heterozygote corresponding to the gene GWT1 really shows the most hypersensitive phenotype to the drug. These are all z-scores, normalized z-scores. So it's most sensitive. It shows a dose response that's nice. The other thing that's nice about this profile is there are 2 other heterozygotes that correspond to gene products that are involved in the preceding step of GPI biosynthesis, to which the GWT1 enzyme acts. So there's biological relevance to a simple profile to really reinforce, one's interest in a compound like that. We also screened, I think, close to 2,500 natural product extracts, so crude mixtures, again demonstrating bioactivity against Candida albicans. So a gemisch of metabolites against the entire strain set. And so here is one of these fitness test profiles that, again, caught our eye because first, the hypersensitivity of the first strain, the most sensitive strain, again, really dramatic depletion of that heterozygote in the pool. But also, again, the biological relevance. Every one of these heterozygotes corresponds to subunits of the COP complex that we heard about just a moment ago. And again, the more you understand about the biology and the function of each of these enzymes, the more you can extract the significance of this profile. So really quickly, MCD4 is also involved in GPI biosynthesis. It's an ER-localized protein, multiple transmembrane domains, and it's retained in the ER by a retention signal at the C-terminus, lysine XX. That series of 5 additional heterozygotes correspond to a machinery that's responsible for the retention and the retrieval of ER membrane proteins like MCD4. And so the biological significance is huge here, where the prediction is you have a target, the most sensitive strain, but each of those other hypersensitive strains are actually demonstrating a phenocopy of the MCD4 heterozygote, right? If you're not retaining the correct localization of MCD4, you're mislocalizing it. It's kind of analogous to only expressing half as much, right? The other thing here is if we think about natural products. So what I'm showing you here is the fitness test profile of the purified compound from the extract. The reason we chased this down is because the crude extract had the exact same profile, okay? And that's important when you're screening natural products. Mike's nodding his head, because that is a profile that is not, you don't see that with known antifungal agents, okay? So you say right there, it's not an echinocandin, it's not rustmycin, it's something unique, right? And so it allows you to really, with confidence, say if we track that down, that's going to be a new compound. So with the 2 preceding profiles I showed, here's the hypothesis, bottom right-hand corner. Just sort of a schematic diagram of what the GPI precursor looks like. And based on our fitness test profiles, we predicted that the natural product inhibitor is affecting the addition of this ethanolamine phosphate on the first mannose precursor, and that our synthetic small molecules were inhibiting acylation of the GPI precursor. And you know that GPI serve as anchors. They're covalently attached to cell wall proteins, cross-linked to beta-1,6 glucan, and these GPI cell surface mounted proteins really sort of coat the surface of the cell wall, and that becomes significant in a moment. Okay, so the fitness test profiles are just a hypothesis, all right? So going back to the principles that I laid out in the beginning, this is what I was talking about in terms of really a robust and rapid means of genetically demonstrating mechanism. So here we've pivoted from Candida albicans to yeast, Baker's yeast, a haploid strain, isolate multiple stable, independently derived, drug-resistant mutants. And you can see at the top here for our natural products, 15 different isolates, whole genome sequencing. This is the sum total of non-synonymous mutations in those 15 genomes. And it's obvious that the hypothesis was met, that in each case we can map an amino acid substitution that maps back to MCD4. And the same for the synthetic compound G884. Only 3 isolates tested in all cases. We're identifying mutations that map back to the predicted drug target. So a very powerful way to just cut to the chase. So to sort of build some sort of biochemical orthogonal mechanism of action data, we reached out to Susan Lindquist and Catherine McClellan at MIT. And Catherine did this experiment here where, I don't know if that shows out very well, but in membrane preps of yeast and a radio-labeled UDP glucanac, she could follow the production of the acylated GPI precursor and demonstrate with our synthetic molecules that we abolish this acylation. The other thing that Catherine did that I thought was really, really nice was in a sort of transgenic yeast system where, in this case, GWT1 is deleted and complemented with either the wild type yeast gene or with the human ortholog to GWT1, this gene called PIGW. She could go back and just look at the MICs, in this case 884, and show quite clearly that there is some real selectivity for the compound against the fungal enzyme versus the human ortholog. Another thing we did was was to demonstrate that inhibitors to 2 different sort of sequential steps in GPI precursor biosynthesis are chemically synergistic, and significantly so. So here, these studies are in Candida albicans. You can see that at 1/4 the MIC of each of these GWT1 inhibitors plus 1/10 the MIC of the MCD4 inhibitor, you get really nice synergy. We also reached out to Charlie Boone and members of his lab at University of Toronto, and what Charlie's team demonstrated was that genetically GWT1 and MCD4 are synthetically lethal, or at least synthetically sick. So they have a really strong genetic interaction between the two, right? And so each of these genes are essential, and so they had to use conditional mutants to each of these 2 genes. But if you dissected tetrads and followed the double mutant, you could see clearly that synthetic sick or synthetic lethal phenotype. And that's really cool because, what this is really a genetic prediction of the synthetic lethal, of the synergistic activity of inhibitors to those two targets. It's not something you see very often, right? And it really clearly demonstrates that the synergy that we see is due to inhibiting those two targets rather than off-target activity, leading to some sort of synergistic effect. Okay, so a few years back, Rob Wheeler and Jerry Fink at MIT published some really interesting papers describing the role of beta-1,3 glucan as a really strong agonist of toll-like receptors to really elicit a pro-inflammatory response, right? Recruit the innate immune system to combat the infection. And what was really interesting about their work was that they built a hypothesis and tested it pretty hard that Candida albicans and other fungi have sort of evolved to sort of mask the beta-1,3 glucan, so that the fungi are hidden from the innate immune system, right? And so, consistent with that, if we stain Candida albicans with an antibody against beta-1,3 glucan, there's no staining, no immunostaining. But at a sub-MIC, an IC40 of any of these GPI inhibitors, we now get some really dramatic staining, right? So we think what we're doing is we're permeabilizing that outer layer of the fungal cell wall so that a big molecule like an antibody can actually penetrate and recognize the glucan. And potentially sort of recruit an innate immune response. And so, consistent with that, it's also true that such drug-treated Candida albicans, the sub-MIC, when co-incubated with macrophages, we could detect between a 2 and a 2.5-fold increase of TNF alpha and other cytokines. So just a nice sort of added sort of value to the mechanism of action of the compound that we could score on a Petri plate. There could be greater bang for your buck. So really quickly, here's just some MIC data on the compounds that I've described. In the case of the GWT1 inhibitors, we get partial spectrum across Candida, some glaring holes. Depending on the GWT1 inhibitor, we can extend the spectrum to Aspergillus somewhat. But more encouraging, the MCD4 inhibitors really showed good potency and good spectrum across key fungal pathogens, right? Really, to the extent of something like caspofungin or amphotericin B. So that's a really nice start. So finally, I'll wrap this up. Obviously one of the questions we had is, could we demonstrate that these compounds are efficacious in a relevant infection model of candidiasis? And here's where a little bit of medicinal chemistry came in, because it turns out that the actual natural product that we isolated is unstable. It has an ester linkage here that is vulnerable to esterases in the plasma. And once you clip off this little tail, the rest of the molecule is inactive. And so with our chemical brothers at Merck, as I like to call them, by changing that ester to a carbamate linkage, they could protect that chain from the esterase, retain full activity, and with that we could then go in and do in vivo efficacy study. And that's shown here on the right, where so in a systemic model of candidiasis, you can see a clear dose response in efficacy, two different methods or conditions for administering the drug. This semi-synthetic natural product is obviously not as efficacious as the gold standard at Merck. We call CanCitis the gold standard. So if this is a home run, maybe this is a base hit. But it is important to point out that in these studies there are no obvious adverse effects. So this is what we'd call a really interesting starting point. And with that, I'll stop and answer any questions you have.